Good morning. Myself Dr. K. Swathi from Sri Padmavati Mahila Vishwavidyalayam, working as Associate Professor in Institute of Pharmaceutical Technology. So today's topic is Analytical Method Validation from Pharmaceutical Validation M.Pharm first sem. Comprehensive steps in method validation, validation parameters as per IP, as per USP. What is validation? Process validation is documented program which provides high degree of assurance that a specific process will consistently produce a desired result or product meeting its predetermined specification and quality characteristics. Analytical method validation. Method validation is the process of establishing a documented evidence which provides high degree of assurance that product will meet the requirements for the intended analytical applications. There are many reasons for the need to validate analytical procedures. Among them are regulatory requirements, good science and quality control requirements. The Code of Federal Regulations 311.165 explicitly states that the accuracy, sensitivity, specificity and reproducibility of test methods employed by the firm shall be established and documented. Of course, as scientists, we could want to apply good science to demonstrate that the analytical method used had demonstrated accuracy, sensitivity, specificity and reproducibility. Finally, management of the quality control unit would definitely want to ensure that the analytical methods that the department uses to release its products are properly validated for its intended use, so the product will be safe for human use. Analytical methods need to be validated, verified or revalidated in the following instances: before initial use in routine testing, when transferred to another laboratory. Types of analytical procedures to be validated. Discussion of the validation of analytical procedures is directed to the four most common types of analytical procedures: identification tests, quantitative test for impurities content, limit test for the control of the impurities, quantitative tests of the active moiety in sample of drug. Steps in method validation. Develop a validation protocol or operating procedure for the validation. Define the application, purpose and scope of the method. Define the performance parameters and acceptance criteria. Define validation experiments. Verify relevant performance characteristics of equipment. Qualify materials, example standards and reagents. Perform pre-validation experiments. Adjust method parameters or acceptance criteria if necessary. Perform full internal validation experiments. Develop SOPs for the executing the method in the routine. Define criteria for revalidation. Define type and frequency of system suitability tests and or analytical quality control checks for the routine. Document validation experiments and results in validation. Validation parameters as per ICH and USP. ICH as per ICH is accuracy, precision, repeatability, intermediate precision, reproducibility, specificity, detection limit, quantitation limit, linearity, range, robustness, system suitability, all should be checked in the ICH. Whereas in the USP, accuracy, precision, specificity, detection limit, quantitation limit, linearity and range, ruggedness and robustness. So in this way, the system suitability and different types of precisions can be missed and detection limit also missed. So coming to the accuracy of an analytical procedure expresses the closeness of agreement between the value which is accepted either as a conventional true value or an accepted reference value and the value found. This is sometimes termed trueness. Accuracy should be established across the specified range of the analytical procedures. Accuracy should be assessed using a minimum of 9 determinations over a minimum of 3 concentration levels covering the specified range. Example, 3 concentrations are 3 replicates each of the total analytical procedure. Accuracy should be reported as percent recovery by the assay of known added amount of analyte in the sample or as the difference between the mean and accepted true value together with the confidence intervals. Precision. The precision of an analytical procedure expresses the closeness of the agreement between the series of measurements obtained from the multiple sampling of the same homogeneous sample under the prescribed conditions. Precision may be considered at 3 levels: repeatability, intermediate precision and reproducibility. Precision should be investigated using homogeneous authentic samples. However, it is not possible to obtain a homogeneous sample. It may be investigated using artificially prepared samples or sample solution. The precision of an analytical procedure is usually expressed as the variance, standard deviation or quotient of variation of series of measurements. Repeatability. Repeatability expresses the precision under the same operating conditions over a short interval of time. Repeatability is also termed intra-assay precision. Repeatability should be assessed using a minimum of 9 determinations covering the specified range for the procedures. A minimum of 6 determinations at 100% of the test concentration. Intermediate precision. Intermediate precision expresses within laboratories variations, different days, different analysts, different equipments, etc. The extent to which intermediate precision should be established depends on the circumstances under which the procedure is intended to be used. The applicant should establish the effects of random events on the precision of the analytical procedure. Typical variations to be studied include days, analysts, equipment, etc. It is not considered necessarily to study these effects individually. The use of an experimental design is encouraged. Reproducibility. Reproducibility expresses the precision between the laboratories. Collaborative studies usually applied to standardization of methodology. Reproducibility is assessed by the means of an interlaboratory trial. Reproducibility should be considered in case of the standardization of an analytical procedure. For instance, for inclusion of procedures in pharmacopoeias. These data are not part of the marketing authorization closure. Specificity or selectivity. Specificity is the ability to assess the unequivocally the analyte in the presence of components which may be expected to be present. Typically, these might include impurities, degradants, matrix, etc. Lack of specificity of an individual analytical procedure may be compensated by other supporting analytical procedures. Detection limit. The detection limit of an individual analytical procedure is the lowest amount of analyte in a sample which can be detected but not necessarily quantitated as an exact value. Several approaches for determining the detection limit are possible depending on whether the procedure is non-instrumental or instrumental. Approaches other than those listed below may be acceptable based on visual evaluation, based on signal to noise, based on the standard deviation of the response and the slope. The detection limit may be expressed as dl equal to 3.3 sigma by s, where sigma is the standard deviation of the response, s is the slope of the calibration curve. The slope s may be estimated from the calibration curve of the analyte. Quantitation limit. The quantitation limit of an individual analytical procedure is the lowest amount of analytical in a sample which can be quantitatively determined with the suitable precision and accuracy. The quantitation limit is parameter of quantitative assays for low levels of compounds in sample matrices and is used particularly for the determination of the impurities and or degradation products. Several approaches for determining the quantitation limit are possible depending on whether the procedure is non-instrumental or instrumental. Approaches other than those listed below may be acceptable based on visual evaluation, based on signal to noise approach, based on the standard deviations of the response and the slope. The quantitation limit may be expressed as QL equal to 10 by S, whereas sigma the standard deviation of the response, S is the slope of the calibrated curve. The slope S may be estimated from the calibration curve of the analyte. Linearity. The linearity of an analytical procedure is its ability to obtain test results which are directly proportional to the concentration of analyte in the sample. Linearity should be evaluated by visual inspection of a plot of signal as a function of analyte concentration or content. If there is a linear relationship, test results should be evaluated by appropriate statistical methods, for example by calculation of a regression line by the method of least squares. The correlation coefficient y-intercept slope of the regression line and the residual sum of squares should be submitted. A plot of the data should be included. In addition, an analytical analysis of the deviation of the actual data points from the regression line may also be helpful for evaluating linearity. For the establishment of linearity, a minimum of 5 concentrations is recommended. Range. The range of an analytical procedure is the interval between the upper and lower concentration of analyte in the sample.